Hi, and welcome to the translation room. The first step in translation is charging. During charging, the tRNA molecules must be chemically linked to their respective amino acids. In the initial step of charging, the amino acid is put into an activated form by reacting with ATP, creating aminoacetyldelic acid. The aminoacetyldelic acid forms and creates a complex with the aminoacetyl tRNA synthase, an enzyme that oversees the process of charging. This new complex will then covalently bond with a certain tRNA molecule. The next step in initiation. During initiation, the small subunit of the ribosome is bound to the initiation, usually 8 base pairs upstream from the Shine-Dalgarno sequence. The Shine-Dalgarno sequence helps align the small subunit of the ribosome to the start codon. Next, the initial tRNA carrying an anticodon binds to the start codon. Usually in prokaryotes, the initial tRNA is carrying the amino acid for methylmethionine, which is the amino acid the start codon calls for. The large subunit of the ribosome then binds to the small subunit encasing the tRNA. GTP, an initiation factor, supplies the required energy for the other initiation factors, F1, IF2, and IF3, to be released. The next step is elongation. In the ribosome, 3 different sites, the A, the P, and the E sites, the tRNA is already bonded to the start codon during initiation, this occupying the P site. During elongation, another charged tRNA moves into the site, binding its anticodon with the mRNA codon. The formethylmethionine is moved to the A site tRNA. The P site tRNA moves to the E site, then exits the ribosome. The ribosome then moves along the mRNA. This process is repeated, forming a polypeptide chain. Elongation continues until a stop codon is reached. The last step of translation is termination. When a stop codon is reached, a release factor is called upon and inhabits an empty A site. The release factor cuts off the polypeptide chain from the translation complex, and the ribosome disconnects and dissociates. Some differences between prokaryotes and eukaryotic translation are in initiation, the initiating codon in eukaryotes is always AUG. In eukaryotes, in contrast with prokaryotes, do not use specific purine-rich sequence in the 5' site of the distinguished initiator, AUGs from the internal ones. Instead, the AUG nearest to the 5' end of mRNA is usually selected at the start site. The 40S ribosome attaches to the cap at the 5' end of eukaryotic mRNA. and this searches for an AUG codon by moving step by step in the 3' direction. The scanning process of eukaryotic protein synthesis is powered by the helicase that hydrolyzes APT. Pairing of the anticodon in met-tRNA with the AUG codon of mRNA signals that the target has been found. In most all cases, eukaryotic mRNA has only one start site and hence is the template for a single protein. In contrast, a prokaryotic mRNA can have multiple Schindel-Garnell sequences and hence start sites. It can serve as a template for the synthesis of several proteins. In elongation termination, eukaryotic elongation factors EF1A and EF1BY are the counterparts of the prokaryotic EFTU and EFTS. The GTP form of EF1A delivers aminoacyl tRNA to the A site of the ribosome, and the EF1BY catalyzes the exchange of GTP found and bound by GDP. Eukaryotic EF2 mediates GTP-driven translocation in much of the same way as does prokaryotic EFG. Termination in eukaryotes is carried out by a single release factor of ERF1 and compared to in prokaryotes. Finally, the ELF3, like its prokaryotic counterpart, IF3, prevents the reassociation of ribosomal subunits in the absence of initiation complex.